Welcome to today's Mercodia webinar with Dr. Jennifer Stern. She will talk about glucagon signaling in obesity and type 2 diabetes. My name is Jenny Presto and I'm the medical science liaison at Mercodia in Sweden, and I will be your moderator today. Dr. Stern is an assistant professor at the Division of Endocrinology at the University of Arizona. Her research is focused on understanding the role of glucoregulatory hormone signaling in obesity, aging, and exercise. Dr. Stern earned a PhD in nutritional biology from the University of California, where she studied caloric restriction and insulin signaling in aging with John J. Ramsay. Her postdoctoral fellowship focused on glucagon signaling in obesity and type 2 diabetes, together with Roger Unger and Philip Scherer at the Touchstone Diabetes Center at UT Southwestern Medical Center. During the webinar, you will be able to write questions to Dr. Stern by typing your question into the question panel of the control panel. Your questions will be only visible for us organizing the meeting, but not for all attendees. After the talk, if time allows, I will read the questions out loud on a first-come, first-served basis for Dr. Stern to answer. Questions that we don't have time to bring up today will be answered through email. It is now a great honor for Mercodia and me to give the word to Dr. Stern for her talk on glucagon signaling in obesity and type 2 diabetes. Please, Jennifer, I leave the word and the platform to you.
Great. As I was saying, thank you, Jenny, and also thank you to Mercodia for inviting me to give this webinar. It's really nice to have the opportunity to interact with the scientific community, especially during these strange times of social isolation. So I apologize if there's a delay in the advancement of my slides. Seems we're having some technical difficulties, but I'll try to keep the pace of my talking with the slides. So as Jenny mentioned, my lab is interested in understanding the role of glucoregulatory hormone signaling in aging and obesity. And we're particularly interested in the role of glucagon. So glucagon is secreted from pancreatic alpha cells in response to low blood glucose or hypoglycemia. And glucagon acts primarily at the liver to promote hepatic glucose output through glycogenolysis and gluconeogenesis in order to maintain normal glycemia during, for example, an extended fast or a bout of exercise. And glucagon counteracts the metabolic actions of insulin, which is secreted from pancreatic beta cells, as you can see here. Insulin acts to promote glucose uptake from peripheral tissues, and it acts to promote glucose metabolism and storage in the form of glycogen and fatty acids through de novo lipogenesis. Now, just as glucagon and insulin are very much interconnected in their maintenance of glucose homeostasis, glucagon is also very tightly connected. The discovery of glucagon is very tightly connected with that of insulin. Now, in 1921, Banting, Best, and colleagues discovered insulin by injecting pancreatic extract into diabetic dogs. And they observed this hypoglycemia, or decrease in blood glucose, in response to injecting the pancreatic extract. Now, what we don't see here in this graph, because each of these hash marks represent 1 hour, is what happens in the minutes immediately after injecting the pancreatic extract. But James Collip made this observation that there was a sharp rise in blood glucose before insulin's induced hypoglycemia. And in 1923, when Kimball and Merlin were attempting to purify insulin, they identified this hyperglycemic substance. You can see here in the dogs injected with pancreatic extract, they showed a rise in blood glucose. And they named this, this substance that had a distinctly hyperglycemic effect, glucagon, which was short for glucose agonist. Now, between 1929 and 1935, a group of German scientists published some very important findings where they identified that the liver is the main target of glucagon action. And they did this by injecting pancreatic extract directly into the hepatic portal vein. They also identified that this hyperglycemic effect was dependent on liver glycogen content. So they showed two very important things. One, that the liver is the main target of glucagon, and that this increase in blood glucose is dependent on glycogenolysis. Now, when glucagon was finally purified in 1949, Earl Sutherland and colleagues showed finally this first spike in glucose in response to injecting pancreatic extract. So these are the data points that we're missing here from this 1921 paper. And then, of course, this was followed by insulin's hypoglycemic effect. Now, when they purified glucagon, they showed a rise in blood glucose after injecting this into rabbits and a return to normoglycemia. So insulin was no longer present because we don't see that hypoglycemic effect. And a few years later, a group of French scientists observed that glucagon is produced in pancreatic alpha cells. They made this observation by treating pancreatic alpha cells with cobalt to inhibit the activity. So you can see here that cells treated with cobalt had a much lower glucagon secretion than did the control alpha cells. Now, the role of insulin in insulin signaling has been extensively studied and very well appreciated for many, many years, but it wasn't until much later that glucagon emerged as a key player in the pathogenesis of type 2 diabetes. And this is one of the first studies that demonstrated that. This came out of my postdoc mentor, Roger Unger's lab, where he and his colleagues demonstrated that in contrast to healthy individuals who respond to a meal with a decrease in circulating glucagon, those individuals with type 2 diabetes are unable to suppress glucagon. And in fact, you can see that they remain hyperglucagonemic even 3 or 4 hours after consuming this meal. Now, it's important to note that since this study and others have come out, it's been demonstrating that this hyperglucagonemia is not only in type 2 diabetic individuals. We also see this in obese individuals who are insulin resistant. So other groups such as Philip Knopp's group have made that observation. And we also made this observation in a cohort of obese individuals who are insulin resistant. In response to a breakfast meal, we saw a rise in glucagon, and they maintained this hyperglucagonemic state compared to the lean control individuals. And if we look at circulating glucagon in the obese mouse, we see that they exhibit the same hyperglucagonemia compared to the lean animals. And even after I inject these animals with a large bolus of glucose, which has a very strong inhibitory effect on glucagon secretion, you can see that they remain hyperglucagonemic compared to the lean control mice. So why is glucagon such a key player in type 2 diabetes? Well, if we think about what happens when we consume a meal, we see a rise in blood glucose. Now, glucose enters into the beta cells, stimulating insulin secretion. And insulin acts in a paracrine fashion on the alpha cell to suppress glucagon secretion. Glucose also enters directly into the alpha cell and can directly inhibit glucagon secretion as well. Now, insulin acts on peripheral tissues such as skeletal muscle and adipose tissue to promote GLUT4-mediated glucose uptake. And insulin also acts on glucose coming into the liver to inhibit gluconeogenesis and glycogenolysis, and it stimulates glycogen synthesis and de novo lipogenesis, keeping that excess glucose in a nice storage form. And so as a result, in the insulin-sensitive healthy individual, we see a decrease in blood glucose and all is well. We're able to maintain euglycemia. In contrast, in the insulin-resistant obese individual, we're no longer able to take glucose up into our peripheral tissues as effectively as a result of insulin resistance. And just as our peripheral tissues become insulin resistant, it's been shown that our alpha cells also become insulin resistant. So we lose this paracrine effect here of insulin on alpha cell glucagon secretion. We also lose insulin's stimulatory effect on glycogen synthesis and de novo lipogenesis, and its inhibitory effect on glycogenolysis and gluconeogenesis. And so as a result, we see an increase in circulating glucagon. It's also been shown that insulin-resistant alpha cells are not able to sense glucose as well. And so with that, we see a further increase in glucagon. And glucagon acts at the liver to promote glycogenolysis and gluconeogenesis. It inhibits glycogen synthesis and de novo lipogenesis. And as a result, we see an increase in hepatic glucose output and hyperglycemia ensues. Now, in fact, it's been shown that suppression of glucagon receptor signaling is very effective in lowering blood glucose. Maureen Charon's group in 2003 was the first to generate a global glucagon receptor knockout mouse. And you can see that they maintain lower blood glucose throughout the day and throughout an oral glucose tolerance test. Dan Drucker and Al Powers went on to generate a liver-specific glucagon receptor knockout mouse, and you can see that this oral glucose tolerance curve is very similar to that of what the Charon group observed in the global knockout. That is, mice lacking glucagon receptor signaling specifically at the liver has significantly lower blood glucose, and they maintain this throughout the oral glucose tolerance test. So this also demonstrates the importance of glucagon receptor signaling at the liver. Roger Unger's group also went on to demonstrate that in a mouse model of type 1 diabetes where streptozotocin is used to ablate beta cells, therefore eliminating insulin, the resulting hyperglycemia can be resolved by eliminating glucagon receptor signaling. And more recently, several clinical trials have come out demonstrating the glucose lowering effects of glucagon receptor antagonists in people with type 2 diabetes. And you can see in this particular study, there's a nice dose-dependent effect on lowering blood glucose. Now, since these studies have come out, more recently, the pharmaceutical industry has really flooded the literature with reports of the safety and efficacy in using glucagon receptor antagonists to lower blood glucose in people with type 2 diabetes. So this is a really great idea, assuming that glucagon is always high in people with type 2 diabetes. But we wanted to understand what happens to glucagon levels in obese, obese insulin-resistant mice in response to fasting and refeeding. So we performed the following study where we fasted lean and obese mice for 4, 8, 16, and 24 hours. And we sacrificed these mice at the end of the fasting period and measured glucagon. And as we would expect in the lean healthy mice, we see a progressive increase in glucagon in the circulation in response to fasting. But what we saw in the obese mice was a stark contrast. As we and others have shown before, obese mice are hyperglucagonemic in the fed state, as you can see here. But as we fast the mice, we see a very robust decrease in circulating glucagon. And in fact, glucagon remains suppressed even 16 and 24 hours after fasting these mice. So we wanted to look at insulin. Now, insulin changes as we would expect. It decreases in both lean and obese mice. But of course, the obese insulin-resistant mice remain hyperinsulinemic compared to lean animals. So what does this mean then in terms of the glucagon to insulin ratio? Well, indicatory of a predominant glucagon signal with fasting and lean mice, we see a progressive rise in this ratio. However, because glucagon and insulin both decrease with fasting in the obese mice, we see no change at all in response to fasting. So we have a very non-responsive glucagon to insulin ratio in obese mice. Now we looked at glucose, and glucose changes or doesn't change as we would expect. It decreases with fasting in lean mice, and again, indicative of an insulin-resistant state. We don't see a robust decrease in serum glucose with fasting in obese mice. So I want to come back to this glucagon to insulin ratio because, as I showed on my first slide, these two hormones are very much related in terms of the maintenance of glucose homeostasis and also lipid homeostasis. So glucagon acts through its G protein coupled receptor by activating the G alpha S proteining, activating adenylate cyclase, which then increases intracellular levels of cyclic AMP and activating protein kinase A. Now, protein kinase A goes on to activate glycogenolysis and gluconeogenesis, so it promotes hepatic glucose output, but it also stimulates fatty acid beta oxidation and inhibits de novo lipogenesis. And glucagon also acts through pKa to regulate cyclic AMP response element binding protein. So at the level of gene transcription, we're also regulating genes that are involved in these metabolic pathways. Now, insulin counteracts the metabolic actions of insulin, primarily through the activation of phosphodiesterase, which promotes the degradation of cyclic AMP and would end these metabolic processes here. So yes, glucagon would increase blood glucose by stimulating glycogenolysis and gluconeogenesis, but glucagon also exerts important effects by stimulating beta oxidation and decreasing de novo lipogenesis. So this begs the question then, are there negative consequences to blocking glucagon receptor signaling? And in fact, many of these clinical trials have reported some negative effects. For example, an increase in liver enzymes, ALT and AST. Now, this could indicate liver injury, potentially as a result of increased glycogen content in the liver. Some studies have also reported an increase in hepatic lipid accumulation, and several studies have also reported elevated cholesterol in patients taking these antagonists. Now, if we look at mouse models of glucagon receptor deletion, for example, Dan Drucker's study in global glucagon receptor knockout mice, he probably could have predicted some of these negative effects in the clinic. So his group showed back in 2008 that global glucagon receptor knockout mice are unable to respond to a fast with an increase in lipid oxidation. So here you can see in wild type mice, we see an increase in palmitate oxidation in the liver in response to a 16-hour fast. And in contrast, global glucagon receptor knockout mice are unable to respond with an increase. And in fact, it looks like there's a decrease. Now, they've also shown that global glucagon receptor knockout mice have an increase in triglyceride secretion from the liver. And Dan Drucker and Al Powers went on to show potential negative effects of blocking glucagon receptor, specifically at the liver. Now, I want to take you back to this figure here. Yes, these animals have a very impressive lowering in blood glucose throughout an oral glucose tolerance test, but they also show severe alpha cell hyperplasia. You can see here, alpha cells are stained in green. These are glucagon positive cells. And you can see compared to the wild type flox control mouse, you have a lot of alpha cell hyperplasia in the hepatocyte specific knockout. And they also reported an increase in pancreas weight. So this could lead to some pretty serious consequences down the road. Now, what about benefits of increasing glucagon receptor signaling? Well, in fact, it's been shown that glucagon receptor agonism can decrease hepatic lipid and decrease plasma cholesterol. Now, these are the opposite effects that we've seen in the global knockout mice and in the clinical studies. Now, this study that came out of Matthias Schopp's group, along with an impressive team of scientists in 2015, tested the effects of an incretin coagonist which consisted of GLP-1 and GIP, compared to a triagonist, which added in glucagon. And here we're looking at livers from mice either treated with the vehicle, treated with the GIP, GLP-1 coagonist, and treated with the triagonist, which included glucagon. And you can see a very nice decrease in hepatic lipid accumulation in mice treated with the triagonist compared to the coagonist. And importantly, they showed that this was independent of changes in body weight. And just earlier this year, another group demonstrated the beneficial effects of agonizing glucagon receptor. You can see here diet-induced obese mice that were treated with the glucagon receptor agonist show a decrease in liver triglycerides. And importantly, they showed that this was independent of leptin receptor signaling at the liver. They also showed a nice decrease in plasma cholesterol in mice treated with the glucagon receptor agonist. So coming back to our liver, we know that fasting in the lean mice and obesity in the fed state leads to a rise in glucagon. And as I've shown, glucagon activates adenylate cyclase, which ultimately leads to an increase in beta oxidation, a decrease in de novo lipogenesis, and an increase in hepatic glucose output. Now, glucagon also acts through this pathway by activating the G-sub-Q protein and activating AMP kinase by phosphorylation. And AMP kinase goes on to also inhibit de novo lipogenesis and stimulate beta oxidation. So we wanted to look at some of these downstream effects of glucagon signaling. Now, coming back to the glucagon to insulin ratio, where we see a rise in this ratio in lean mice as we progressively fast them for an extended period of time, and a non-responsive ratio in the obese mice. So first, we looked at liver cyclic AMP, and we found that liver cyclic AMP content increased very similarly as did the glucagon to insulin ratio in lean mice. Now, in contrast, obese mice that have a non-responsive glucagon to insulin ratio with fasting show no change in liver cyclic AMP content as we fast them for progressively longer periods of time. Now, if we go one step further and we look at liver glycogen depletion with fasting, you can see that liver glycogen is nearly completely depleted in lean mice after just 16 hours of fasting, and it remains depleted after 24 hours of fasting. Now, in contrast, obese mice show a very much attenuated liver glycogen depletion in response to fasting. And it's interesting because this is despite the fact that they have very similar glycogen levels under fasting conditions. So we next wanted to look at the relationship between glucagon and insulin and hepatic cyclic AMP concentrations in lean and obese mice. So we regressed hepatic cyclic AMP against glucagon and insulin, and we found, as one would expect, in lean mice, as glucagon in the circulation goes up, hepatic cyclic AMP concentration in the liver also goes up. And as insulin concentrations go up, hepatic cyclic AMP concentrations in the liver go down. But when we looked at this relationship in obese mice, we found absolutely no relationship between either glucagon or insulin and hepatic cyclic AMP concentrations. So this leads us to conclude that perhaps hormone-induced changes in cyclic AMP are also dysregulated in obesity. So it's not just what we're seeing in the circulation, but also downstream of both glucagon and insulin signaling. So we wanted to next look at cyclic AMP responsive genes. And we looked at PepCK, glucose 6-phosphatase, and tyrosine aminotransferase, which are genes involved in gluconeogenesis, and also carnitine palmitoyltransferase 1, the rate-limiting step in fatty acid beta oxidation in the liver. We found, again, very consistent with a rise in the glucagon to insulin ratio. Lean mice show an increase in the expression of all of these genes with an increase in fasting duration, yet obese mice show little to no response in the expression of these genes with fasting. So we next asked the question, could we restore the hyperglucagonemia seen in the fed state in obese mice by simply refeeding them for a couple hours? So what we found is, yes, in fact, as you fast an obese mouse for 16 hours, glucagon goes down again. So we repeated that finding, and refeeding them showed a robust increase in glucagon. And in fact, it looks like we've restored this hyperglucagonemia that we found in the fed state. Now, in contrast, we see a rise in glucagon in response to fasting. And interestingly, we see a very slight increase in glucagon secretion with this acute refeeding. If we look further postprandially, glucagon does go down. So this was an interesting finding that we weren't expecting in lean mice. Now, insulin changes just as we would expect. It decreases with fasting and increases with refeeding in lean mice, and we see the same pattern in obese mice. Now, if we go back to that ever important glucagon to insulin ratio, we found, as we would expect, this ratio increases with fasting and decreases with refeeding in lean mice, yet we see no change in this ratio in obese mice. And again, we looked at glucose and we found that it decreases with fasting and increases with refeeding as we would expect in lean mice, and remains pretty elevated throughout the study in obese mice. Again, this is indicative of an insulin-resistant state. So the question is why? Why are we seeing this fasting-induced decrease in serum glucagon and this non-responsive glucagon to insulin ratio in obesity? So we looked at potential paracrine actions that would occur in the pancreatic islet. And here I'm showing you immunofluorescence image of a pancreatic islet from a mouse. You can see the majority of the islet consists of insulin positive beta cells. You can see here stained in red. And the outer portion of the islet is studded with glucagon positive alpha cells. And somatostatin positive delta cells are sort of scattered throughout the islet. So insulin secreted from the beta cell exerts a paracrine fashion on alpha cell glucagon secretion. Somatostatin secreted from the delta cell also exerts an inhibitory effect on both beta cell and alpha cell insulin and glucagon secretion, respectively. So we wondered, are we perhaps temporarily insulin sensitizing the alpha cell by fasting these obese mice so that the alpha cell is better able to respond to insulin with a decrease in glucagon secretion? And I want to share with you a pretty exciting study that came out of a group from the Joslin Diabetes Center, where they deleted the insulin receptor from the alpha cell. And they demonstrated that fed glucagon is significantly elevated regardless of age without insulin receptor signaling at the alpha cell. Now, they went on to perform a perfusion study where they perfused the pancreas of these mice and gradually increased the concentration of glucose in the perfusion buffer. And they observed the insulin response. Insulin increased equally in all groups. However, if we look at glucagon secretion throughout this study, they found that alpha cell insulin receptor knockout mice had significantly higher glucagon secretion. So we performed the following perfusion study that was a little bit different from this group. We wanted to look at low glucose conditions. So we kept glucose low at 2 millimolar after a brief acclimation period where 6 millimolar glucose was perfused into the mouse. And during this period of time, we increased the concentration of insulin in the perfusion buffer from no insulin to insulin concentration similar to what we see in the serum of a lean fasted mouse, as I've shown here, a lean fed mouse, an obese hyperinsulinemic mouse, and then supraphysiological levels of insulin. And so right off the bat, we noticed, and I'm sure you are too, that under low insulin, no insulin, and low glucose, Mice that have been fasted for 16 hours have a very robust decrease in glucagon secretion compared to obese fed mice. So this was a really nice observation because it ruled out the possibility that perhaps fasting an obese mouse somehow increases glucagon clearance. We know this is directly pancreatic glucagon secretion. Now we added in a lean-fed control group, as you can see here, and I want to draw your attention first to these first 10 minutes of the study where no insulin was present, but we see a shift from 6 millimolar to 2 millimolar glucose in the perfusion buffer. And we found that lean-fed mice respond to this shift to hypoglycemia with a very robust rise in glucagon secretion. However, both groups of obese mice, regardless of being fasted or fed, do not respond to low glucose with an increase in glucagon secretion. Now, next, if we look at this next 10-minute period where we added a very low concentration of insulin to the perfusion buffer, we found that lean-fed mice have a very robust decrease in glucagon secretion. Obese fed mice do respond to this shift to insulin, but it's quite low, only 13% change in glucagon secretion. Yet obese fasted mice show an increase in this glucagon response. So this suggests that perhaps we may be insulin sensitizing the alpha cell temporarily by fasting mice for an extended period of time. They're better able to respond to insulin with a decrease in glucagon secretion. So next we looked at somatostatin because somatostatin inhibits both beta cell and alpha cell insulin and glucagon secretion respectively. And we found right off the bat that lean-fed mice had the highest amount of somatostatin secretion, and obese-fed mice had the lowest somatostatin secretion. Fasting an obese mouse increased somatostatin significantly. So this could be potentially another mechanism by which fasting decreases both insulin and glucagon secretion in the obese mouse. Now, the response to insulin in these mice is identical if we plot this data as a percent change in somatostatin from the perfusate. Now, I want to take you back to our mouse islet and compare this with a human islet. Now, it's important for all of us basic scientists to remember that we are aiming to treat obesity and type 2 diabetes in humans. So we want to see if our findings at the bench can translate to the clinic. Now, you'll notice that the human islet is organized very differently from that of a mouse. Where the mouse has predominantly beta cells, we see a far greater percentage of these green glucagon positive alpha cells in the human islet. And we don't see the same type of organization that we see in the mouse islet. So these cells, including somatostatin positive delta cells, are all interspersed throughout. Now, to understand the effects of fasting and refeeding in obese humans, we've collaborated with Sam Klein at Washington University, and he performed a really exciting set of experiments where he brought obese insulin resistant, again, these were not diabetic individuals, but obese insulin resistant individuals. He brought these individuals into the clinic the night before the study began. They underwent a 12-hour fast after a standardized dinner meal, and then they were fed 3 meals throughout a 24-hour period: breakfast, lunch, and dinner. And you'll notice right off the bat that obese individuals have significantly higher circulating glucagon throughout the study period compared to lean. But I want to focus in here on these first 6 hours after consuming a breakfast meal after an overnight fast. And I want to highlight also that there's a very big difference in fasting duration between mice and humans. So a 12-hour overnight fast in humans really translates to what would be a 4-hour fast in mice. if we wanted to really compare our results in the mouse, looking at the effects of an 8, 16, and 24-hour fast, we would need to fast these humans for upwards of 48, 24, 48, even 72 hours. So we're not recapitulating our study in the mouse, but this does give us a nice idea of how glucagon changes with fasting and feeding in obese individuals. So if we look at this first 6 hours, again, glucagon is higher in obese individuals than lean. And you can see that they respond to a meal with a rise in glucagon, and glucagon stays elevated for a couple hours and then it comes back down to basal levels. Now, in contrast, we really see no significant change in glucagon throughout this 6-hour period in lean individuals. And if we look at insulin, insulin changes as we would expect. It rises and falls with feeding and postprandially in both groups of people. But of course, the obese insulin resistant individuals have higher insulin than do the lean. Now, I want to come back to this glucagon to insulin ratio. And you'll notice that both groups of people do show a decrease in the ratio with feeding, and then a slight increase back to baseline with postprandially. But you'll notice that this is much more dramatic in the lean individuals. So there is somewhat of a blunting in this change in the glucagon to insulin ratio in obese people. And we checked glucose. Glucose changes as we would expect. It rises and falls with feeding and postprandially in both groups of people. Of course, this is higher in the obese insulin resistant group. So what we've concluded from these studies, along with what other groups have shown, is that perhaps we need to rethink glucagon in obesity. We've shown for the first time in the mouse a paradoxical response to fasting, changes in glucagon. And so we see an increase with feeding and a decrease with fasting. And more importantly, this translates to a less responsive glucagon to insulin ratio. And so with that, I'll leave everybody with a question that I can't necessarily answer, but I think it's an important one for us all to consider is, should we inhibit or stimulate glucagon? Of course, there are benefits to inhibiting glucagon receptor signaling. It's very effective in lowering blood glucose in people with type 2 diabetes, but stimulating glucagon is also important in maintaining lipid homeostasis, particularly in the liver. And this is something we also need to consider when we look at fasting-induced changes in the glucagon to insulin ratio. So with that, I'd be happy to take questions, but first I need to acknowledge some very important groups of people. First and foremost, my lab at University of Arizona, all of our wonderful collaborators throughout the country and the world, and my funding through National Institute on Aging and University of Arizona Research Discovery and Innovation Group. And thank you especially to Mercodia for this opportunity and to the audience for your attention.